Video series. Whenever a pathogen enters the human beings, now this elicits two types of immune response. The first one is cell mediated, the other one is humoral mediated immunity. Cell mediated immune response is mainly executed by T lymphocytes. Now these T lymphocytes release variety of cytokines, and these include interleukins, interferons and tumor necrosis factor alpha beta, which will bring about the pathogen destruction. Now, these interleukins are mainly responsible for communication between cells, and these include interleukin 2, which will stimulate proliferation of T cells. Interleukin 4 stimulates B cells to produce antibodies. Interleukin 5 stimulates eosinophils. Interleukin 6 stimulates the release of various acute phase proteins. Interferons are of three types: alpha, beta, gamma. Alpha will stimulate the macrophages towards phagocytosis. Interferons beta are responsible for addressing viruses. Interleukin gamma is responsible for cell differentiation. Tumor necrosis factor alpha and beta is there, of which alpha is of great importance, also known as cacectin. It is stimulator of release of inflammatory biomolecules. It will stimulate programmed cell death, that is apoptosis, and also induces fever. Now, when all these are cumulatively referred to as cytokines, and these cytokines bring about the following effects. One is they provide protection against infections, be it a bacteria or the virus. They are involved in tumor cell destruction, especially the tumor necrosis factor alpha. Now, this cell-mediated immunity is also responsible for delayed type of hypersensitive reaction. So, this refers to overreaction of the immune system resulting in unwanted tissue destruction. The good example is development of erythematous lesion at the site of BCG injection after 48 to 72 hours. So because it will take this much of time, after 48 to 72 hours, it is known as delayed type of hypersensitive reaction. Apart from that, cell-mediated immunity is also responsible for rejection of allograft. Now, the main rationale behind this is as follows. We have humans have major histocompatibility complex gene located on chromosome 6 with A, B, C, D and DR loci. Now, this entire complex is responsible for recognizing self proteins from the non-self proteins or the non-self antigen. Now, this gene with these loci A, B, C, D, DR loci produces around 150 alleles. So enormous different antigens are produced in one particular individual. Now this this antigenic constitution differs from one person to another person. So whenever there is a need for transplantation of organs or tissues, this HLA typing has to be done. So only when the HLA typing gets matched between two persons, then organ transplantation can be done. So if there is no matching between two individuals, what happens? It results in graft rejection. So there are three types of allograft rejection. One is hyperacute rejection, which occurs within minutes to hours once the organ or the tissue is transplanted. Now this is because of the presence of preformed antibodies against the donor antigen. What happens is there occurs rapid thrombus clot formation followed by ischemia and graft failure. The next one is acute rejection, which takes days to months, and this is T cell mediated response wherein the graft gets inflamed and becomes non-functional. The third type of rejection is chronic rejection, characterized, and this takes months to years for the allograft to be rejected. So this is characterized by chronic immune injury to the graft followed by fibrosis and vascular changes. So this is about major histocompatibility complex and the need for going for HLA typing before any organ transplantation or bone marrow transplantation. Coming to types of cell-mediated immunity, there are two types of T cells. One is the T helper cells, which stimulate the production of suppressor T cells and also stimulates the plasma cells to produce antibodies. And we have suppressor T cells, which will inhibit both the helper T cells and the plasma cells, so that there is no production of antibody. Coming to specific characteristic feature of helper T cells, they carry CD4 determinant on their surface. They constitute 50% of the T cell population. Main function is for optimal antibody production because they stimulate plasma cells and these plasma cells in turn produce antibody. Now these helper T cells are selectively destroyed in AIDS. advanced immunodeficiency syndrome. Coming to suppressor T cells, they carry CD8 determinant on their surface. They constitute only 10% of the cell-mediated immunity. Main function is they downregulate the activity of T helper cells and they downregulate B cells so that there is no stimulation of the plasma cells to produce antibodies. And these suppressor T cells are decreased in autoimmune diseases and different types of cancer. Was all about cell mediated immunity. So thank you. Next video will be on humoral immunity. Please do like, share and subscribe. Happy learning.